So in this white part, you draw a complete bean seed. You draw a complete bean seed, just excuse me. This one I mean, you draw this one. Is it all right? Any one. Either this, you draw this one, the first one. This diagram you draw. Is it clear? A complete bean seed. And then you will draw the LS of a bean seed. That means you open the bean seed, you remove the cover of the bean seed, and what you see inside, I mean, you will show this one. You will show this one. In this diagram, here you are showing hilum, you are showing micropyle, but here you are not showing micropyle and hilum. Is it? You are not showing. What are you showing? You are showing radicle, you are showing plumule, you are showing hypocotyle, epicotyle, testa tegmen, then cotyledons. These all things you have to show. Is it all right? Like I mean to say, when you are opening the bean seed, what do you see? You are opening the bean seed, what do you see inside? There you don't see micropyle and hilum. Now what do you write? You write the date first here, 28/5/2020. Now what do you write? To study the structure of a bean seed. All right? Okay, this is the second one. First one was the flower. What do you take? A soaked bean seed is taken. You, instead of soaked, you can write a fresh bean seed is taken. Is it clear? That means you have just taken it out from the bean seed. Okay, not the dried one. If you are taking a dried one, that should be soaked. The parts of the flower, the parts present are as follows. The parts present in the bean seed are as follows. Is it clear? The first one you write seed coat. Structure, you will write how does the seed coat look like. Color is brown in color. Is it clear? And it's thin brown in color. Function, what is the function? It protects the seed. All right. Micropyle structure, it is an opening, tiny opening. What is the function? There are two functions we have done: diffusion of gases, entry of water. Two functions you will write. Third is hilum. Structure you will write, it is a white scar, and then you don't have to write a function, or you can also write it showed the attachment of the seed to the fruit wall. You can write that. So, but you know, later on once it is separated, there is no such function. So hilum you will write, cotyledon you will write, two fleshy structure. Number two fleshy structure. What is the function? Protect, it gives provides nourishment to the seed, and the second one it gives gives nourishment to the embryo. Second one you will write, it provides protection to the embryo. And the fifth one, embryo, you will write first part is plumule, second part is radicle. What is the function of plumule? Give rise to the shoot system. What is the function of radicle? Give rise to the root system. So this is what you have to do for the bean seed. Now we come to the third one, what you can do. These are all available in market right now. Bean seed, if bean is not available, you can soak up little 2-3 rajmas and you can see the things. Now maize is available in market. To study the structure of maize seed, a soaked or a fresh maize seed is taken. The parts present are as follows. The parts in the maize seed present as follows. Date should be given as 28/05/2020. Is it all right? So parts, the first part, outer covering, the fused fruit wall and the seed coat. Structure, it is thin, scaly. Is it clear? Thin. Transparent. Function, protection. Endosperm, fleshy, major part of the seed is occupied by that. And what is the function? Provides nourishment. Scutellum holds embryo. It is not, it provides nourishment, but scutellum, you remember, it is cotyledon, very tiny in maize seed. It is not the main one to provide nourishment to the main seed. Maize seed is albuminous plant. So you remember, endosperm, scutellum, it gives protection to the embryo. Don't write provides nourishment, because providing nourishment is done by endosperm. Coleoptile, you know, leaf-like structure, it protects the plumule. Coleorhiza, leaf-like structure, protects the radicle. Embryo, radicle, embryo two parts, radicle, first one embryo has two parts, the radicle gives rise to root system, plumule gives rise to shoot system. And here what you do, draw the LS of a maize seed, label all the paths shown in your book. Which diagram you are going to draw? This one you are going to draw, this one. This part, when you open up a maize seed, this part is so tiny that it is not very clearly visible. The embryo in monocot are very small. The embryo part is very small, it's not very tiny as they are so prominent in dicot, they are not that prominent in monocot. Is it clear? So draw the diagram of this. You don't have to mention the function of alluron layer or the function of epithelium. Is it all right? The parts which are shown, I have shown 6 parts: the fused fruit wall and seed coat, endosperm, scutellum, coleoptile, coleorhiza and embryo. These parts you will write the function, and this diagram has to be drawn. That's all about this chapter. Thank you, children.